All right, in this video, we are going to address the topic of DNA replication. And so we're going to answer this first question here. How does DNA copy itself? So here we go. Before we can really talk about that, we have to review the structure of DNA. So we already talked about this, but let's review. You guys already know that DNA has this double helix structure, so like a twisted ladder. So that's what this is showing right here. There's a twisted ladder double helix structure to DNA. We also said that DNA is made up of nucleotides, and that nucleotides have three main parts. There's this deoxyribose sugar, a phosphate group, and a nitrogen base. In the picture and in most pictures that you see, the deoxyribose sugar and the phosphate group make up what we call the backbone. So you'll see pictures of DNA that just look like this. Just know that the backbone of the DNA, this blue part right here, is the phosphate group and the deoxyribose sugar. The nitrogen bases are the things in the middle. And we also said that the nitrogen bases follow these base pairing rules. So we said A always goes with T and G always goes with C. And that always is true. It never really changes. Okay, so that's the structure for DNA. So DNA replication. So first of all, the purpose of DNA replication is to make an exact copy of DNA. DNA replication happens pretty much all the time, actually. Anytime a new body cell is made. So this includes like growing and developing, like what you guys are doing, because you're teenagers and you're growing and developing. And then if there's ever repair of body tissue, so if you get a cut on your hand and your skin has to repair itself, DNA has to replicate for when the new skin cells are being made. And then honestly, I didn't include it here, but your cells are constantly dying and then being reborn again, or where cells die and then more cells are made to replace them. So this is actually happening all the time. And essentially, every time a new cell is made, we have to copy the DNA because every cell in the body has to have the same exact copy of DNA. Okay, so how does it happen? That's what we're going to talk about today. All right, so how does DNA replicate itself? Well, first of all, to replicate means to make an exact copy or an exact replica. So that's what it's doing. It's going to be making an exact replica or an exact copy of itself. Just to make it super simple, there's three main steps to this process. The first step is the double helix shape becomes unzipped. And the thing that unzips it is this enzyme called helicase. So that's step one. The DNA, the structure has to become unzipped. Okay, second thing, another enzyme, so once it's unzipped, another enzyme named DNA polymerase is going to join individual nucleotides to both of the strands, okay? And then in doing that, it's going to have two identical DNA strands at the end. After it does that, DNA polymerase is going to proofread its work to make sure that no mistakes were made. Because in replicating the DNA, it's going to go to all the cells in the body, and it would be really bad if there would be a lot of mistakes to your DNA, because essentially that would change who you are. Okay, so this last step is really important. DNA polymerase is going to proofread, make sure that all the nucleotides that he added to the two DNA strands are actually correct. Okay, so if that didn't make sense, here's a picture of it. All cells are going to start out with this original DNA strand. Okay, so this is our starting point. Well, in step one, the strand gets unzipped. Okay, so it's going to get unzipped right down the middle. So these nucleotides are now exposed. Well, DNA polymerase is going to bring in new nucleotides, and he's going to attach them to the exposed strands. Okay, so here, that's there. And then he's going to attach them to this strand as well here. So the result of doing that then is going to be two identical strands at the end. And you can see that this strand here is the same as this strand here, which is the same as the original strand. So you start out with one, it gets unzipped, the nucleotides get added, and in the process, the end result is that you have two identical strands. Okay, so two exact copies or replicas. The reason why this works and why there's two exact exact replicas is because DNA polymerase follows these base pairing rules. So A's are always going to go with T's and C's always go with G's. In doing that, we ensure that the two copies at the end are identical. Because every time you add a new strand, it's going to be complementary. And there's no possible way, like you're not going to have a random N here. It's always going to be A with T, C with G. So you always have the same exact copy at the very end. There's this other word here. We say that DNA replication is said to be like semi-conservative. Translation means kind of conservative. So in this sense, conserve here means to save or recycle. And we're saying that the two strands of DNA at the end, so the two new strands of DNA, both include half of the original nucleotides from the original strand. So the original strand was kind of conserved. So let's look back at this picture to see what that really means. If you look at the two new strands, the green is showing the original strand, and this strand has half the nucleotides from the original strand over here, and then this new strand also has half the nucleotides from the original strand. So we're going to say that this original DNA strand was kind of conserved. So the new strands have part of the original strand to it. Okay, and then lastly, we have this animation to show you. So I'll pull it up here. Okay. Okay. And this basically just shows the process a little bit faster with just a little bit more detail. Here we go. Hopefully you can hear it.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells.
So here's the original strand.
You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left.
Right here.
The whirling blue molecular machine is called helicase.
This is helicase right
It
here.
spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right.
So it's an exact replica.
Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
Okay, cool. So hopefully with the definition and the pictures and the animation, you guys have a good grasp on this stuff. This video will be up. I suggest that you keep the notes that you took today and use those to study for your test. Thanks for watching.